multiple groups. Data using the line graph. Line graph is primarily used to measure data over a period of time, so change over time, minutes, days, hours, etc. These are line graphs that we might see. Scatter plots. I just stole this from our project that we did together, by the way. Make sure you plot every data point that's given to you. After that, draw a best fit line. The line doesn't have to be touching all the data points, but a majority of them is recommended. Try to get as many on top as many on bottom when you're drawing the best fit line. You're looking at pairs of numbers. Maybe you're figuring out how temperature affects plant growth. You would put how tall the plant was on the y-axis and the temperature on the x, or vice versa. Scatter plots are all quantitative. Bar graphs. You'd use a bar graph in biology, compare data from different study groups. Remember categories. We're thinking categories with bar graphs. Use a key if you have different types of bars. Label the axes always for any graph. Make sure you create a scale. Make sure a scale it fits on the whole graph. We were talking about that yesterday with one of the practice FRQs. If your graph is really squatty on the graph paper that they give you, make sure the scale is kind of equalized. Histograms. This is when we shove these together. You can't rearrange the categories of histogram. You can do a bar graph squished together like a histogram, though. That's okay. This is more like shear frequency at intervals of relationships. Okay, your numbers values. It will tell you on the grid-ins how many it wants you to round to. Make sure you know the difference between tenths, hundredths, thousandths, and 10,000ths. If you feel more comfortable with a fraction, just use a fraction because they accept those as well. Okay, these are some of the formulas that we will see on our formula sheet. Make sure you're able to apply standard error, mean, standard deviation, chi-square, definitely chi-square. Remind me, how do we get our degrees of freedom? N-1, number of categories, -1. Very good. So if we have 5 categories or 5 classes that we're looking at, we have what degrees of freedom? 4. What critical value, when we get our critical value, what p-value do we want to look at to see if it's statistically significant? 0.05. So if we had 4 degrees of freedom, 0.05, we would look at that particular number on our table. Okay. Error bars. Make sure you know how to read a graph with error bars and know what the difference is between something that's statistically significant within the error bars and something that is not. If those error bars cross, you don't see its statistical significance. Okay. Surface area and volume. You may be asked to calculate the surface area to volume ratio. It will give you volume of a sphere, volume of a cube, surface area of a cube. All these things, as long as you can plug it in and you can figure out your ratio, you're good there. Rate and growth. We did this very recently, so hopefully this is fresh in your mind. But these particular equations are on here. Do some practice problems if you don't remember the carrying capacity stuff. Water potential is also on there. We did this recently as well. Remember that to get your solute potential, you're going to get negative ICRT. Most of those values are given to you. The C is the only one. That's our molar concentration that you'll have to figure out from the problem. Most of the time I will be 1 because for sucrose, it's 1 because sucrose does not ionize in water, and most of the time we're going to be dealing with sucrose when we deal with these problems. If not, they'll give you another value. R is a constant. Make sure your temperature is in Kelvin, and you can plug it in from there. Just be familiar with that one. Simpson index. Sometimes they'll give you formulas that aren't on your formula sheet, and you'll be able to plug it in. You don't have to memorize the Simpson diversity index, though. But we practice with that. We sum up all the values, and we're good there. Hardy-Weinberg, remember the conditions are large populations, no mutation, no migration, no natural selection, and we have random mating. This is that evolution is occurring or not occurring?
Not occurring.
Not occurring. If your values don't match what you got in the Hardy-Weinberg equilibrium, what can you maybe conclude?
It's evolving.
There is evolution occurring. Good. All right, so big idea 1. Evolution. We've got Darwin. We've got evolution is a change in the genetic makeup of a population over time with natural selection as its major driving mechanism. Make sure you know examples of natural selection. Darwin's theory is supported by evidence from many scientific disciplines. So now we have this modern synthesis theory of evolution where we have all these evidence pieces from biochemistry, from fossils, from embryology, and we can combine that together to look at our... Darwin didn't know about genes and stuff, but he was able to create a natural selection theory. Remember that meiosis and fertilization provides many phenotypes and offspring on which natural selection operates. So we have, the more variation we have, the more phenotypes we're going to have in the population. Good example of natural selection is antibiotic resistance in bacteria. What's another good example of natural selection? That darn peppered moth, yeah. But make sure you have a couple of those examples on hand. Remember, fitness is a measure of evolutionary success, the number of surviving offspring left to produce the next generation. So you are not fit because you are strong, you are fit because you are reproductively successful. Even if you live a long time, if you don't have any kids, are you considered evolutionarily or biologically fit? No. Okay. A diverse gene pool is vital to the survival of a species because environmental conditions change. Maybe at one point you're really fit for one environment, but if that environment changes because of a natural disaster, because of human impact, because of whatever, we can have really disastrous implications if everybody in the gene pool is the same. Diversity is good. Individuals don't evolve. What evolves?
Population.
Populations. Good. Oops, this is repeating. Okay. So don't forget, evolution is a population level phenomenon. The evolution of a population emerges from the individual fitness of members of that population. Alleles to the next generation will change accordingly in natural selection. Okay, so humans can impact evolution as well. We have artificial selection within dog breeds, within food. Bananas are a good example of that. Things that lead to the loss of genetic diversity. Inbreeding. If we have a small population size, if we don't have random mating, if we don't have any migration, so no new individuals coming in or leaving and net lack of mutations. Phenotypic variations are not directed by the environment, so they occur through random changes in DNA through new gene combinations. Okay, so some good examples to keep in mind. DDT case, when we're killing mosquitoes, but then they become unaffected. Sickle cell anemia is when we actually have a selective advantage for the heterozygous condition. If you're just a carrier, you may be resistant to malaria, but you also won't have as bad as symptoms as somebody with sickle cell. But unfortunately, you could marry someone else who's a carrier, and then your child could have sickle cell as well. So it causes a genetic disease, but it can also protect against an infectious disease. Phylogenetic trees, make sure you know how to read and use them. They're also called cladograms. Speciation can be fast or slow. Development of reproductive isolation may take millions of years or may develop in a few generations. What's that called when we have like a phenomenon where all of a sudden a lot of species can develop?
Adaptive radiation.
Adaptive radiation, great. Okay, of these, make sure you know allopatric and sympatric. Allopatric is when there's a physical barrier and then new species develop from there. Sympatric is we have reproductive isolation, but they're in the same area for whatever reason. Organisms may share conserved core processes and features that evolved and are widely distributed among or many organisms today. What is one that they always ask about that is conserved process? Glycolysis. You guys remember that? Some more evidence. Through the process of transformation, genetic information from an organism can change the genotype and phenotype of another organism, thus providing additional evidence for the universality of genetic code. Because of the fact that we can actually do or make genetically modified organisms tells us that the machinery between all organisms is the same. As long as we give the instructions for one gene, we can stick those instructions in another organism. That organism can successfully produce that protein. That means that we have some sort of evolutionary commonalities. All right. So, certain similarities, genetic code, like I said, metabolic pathways, glycolysis, essentially every cell on the planet. Morphological similarities, all cells on the planet are structured on one of the two major organizational plans. What are those two plans? The two types of cells?
Oh, prokaryotic.
Prokaryotic and eukaryotic. And then, of course, endosymbiosis, the theory that major eukaryotic organelles, mitochondria and chloroplasts, evolved from free-living prokaryotic cells and that were engulfed and internalized by eukaryotic ancestors. So we did some experiments for the conditions that existed. We tried to replicate the pre-life conditions on Earth. Miller and Urey did that, and they actually formed sugars, amino acids, and nucleic acids. RNA world hypothesis, the hypothesis that RNA preceded DNA. RNA's double function as a catalyst and as a storage molecule for information. Remember what vestigial structures are. It's one of our evidence for evolution. Earth originated 4.6 billion years ago. Remember the key points in the history of life. Remember that plants evolved before most of our terrestrial animals. We don't have vertebrates for a really, really long time. Just sort of the major extinctions, be aware of their order and kind of what happens when. Okay, big idea too. We're getting there. Free energy, this is what this one's all about. This graph is talking about how an enzyme, its activity can lower the activation energy of a reaction. Water properties, water is polar, so it has positive and negative charges, forms hydrogen bonds. Hydrogen bonds are weak, but when they're all together, they can be strong. There's cohesion, adhesion, high specific heat, high surface tension, capillary action, develops many substances. Surface area to volume ratio, why does this, what do we talk about this? Why do we talk about this? Yay! Jonas.
Large surface area is good because it means that many substances can get into or out of the cell, but and a smaller volume is good so they can get to their specified location.
Good. As we have a larger volume, that ratio will decrease, and so it's harder for things to diffuse and things to do their processes that they're supposed to do. Keep in mind that the greater surface area to volume ratio we have, the more efficient the cell process is. That's why we have limits to cell size. Okay, why organelles? They help with compartmentalization. They provide a favorable local environment for metabolic reactions. They can protect the cell from potentially damaging metabolic reactions. So this compartmentalization gives us these two benefits. And getting things in and out. So viruses or bacteria can be engulfed by cells. We call that phagocytosis, and then they're digested, and then they're excreted through exocytosis. Cellular respiration. We've got three main parts: our glycolysis, our Krebs cycle, and our electron transport chain with oxidative phosphorylation. Be familiar with those. Be sure you can write the general chemical formula, not necessarily the numbers and all the coefficients, but just know what goes in, what goes out to the large process. Make sure you're familiar with what happens in glycolysis, what happens in the Krebs cycle, or also citric acid cycle and oxidative phosphorylation. Remember that glucose is oxidized, and that in our reduction we have something accepting electrons. In oxidation we have something donating electrons. And then those electrons are used in the electron transport chain to form our gradient to help process the formation of ATP. Phew, okay. All right, so these are each of the things that happens in each of the steps. We have NADH produced in glycolysis for use in the electron transport chain. Acetyl coenzyme A for entering the Krebs cycle provides energy a little bit for phosphorylation of ADP. Oxidative intermediates of the Krebs cycle are, we get NADH and FADH2. We get higher energy electrons for use in the electron transport chain, and we get energy to pump protons against their concentration gradient, and GTP for the phosphorylation. And then finally, our proton gradient in the electron transport chain, flow of protons through that ATP synthase is going to generate ATP, or in the large amount of ATP. Okay, glycolysis, one more time. Nearly all existing organisms perform glycolysis. Glycolysis occurs under anaerobic conditions, and glycolysis occurs in the cytosol. All of this is evidence that can point to what?
Common ancestor.
Common ancestor. It's a conserved process. Good. You guys can do this. Okay, photosynthesis. Process that converts solar energy into chemical energy. We've got light energy converted, carbon is fixed into organic compounds. That's the broad summary of it, but you need to break it down into parts. Remember we had that really annoying Hamilton song that you can go back and watch if you remember. We have light-dependent reactions, light-independent reactions. Know what happens in each of the stages and where they happen. Chloroplasts are structurally similar to and likely evolved from photosynthetic bacteria, just like mitochondria. The structural organization of these cells allows for chemical reactions of photosynthesis. Water is important because chloroplasts split water into hydrogen and oxygen, incorporating the electrons into sugar molecules and releasing oxygen as a byproduct. And photosynthesis reverses the direction of electron flow compared to respiration. Photosynthesis is a redox process. Make sure you know the equation as well. Don't worry so much about the numbers, but know what goes in which side. So photosynthesis consists of the light reactions, the photo part, and the Calvin cycle, which is the synthesis part. The pigments are the substances that absorb light. Different pigments absorb different wavelengths, light wavelengths that are not absorbed or reflective, and so that's the color that we see in the green leaves. All right, so again, make sure you know photosystems 1 and photosystems 2 are part of which process. Calvin cycle in general, we consume the carbon dioxide in the Calvin cycle. And we have, again, our summary of photosynthesis. Remember what comes in, what comes out of each part of the process. We have different types of photosynthesis as well. We have C3, and we have different types of plants using these in photorespiration, such as our grasses, such as our cacti. And remember, like, the conditions that we would need for those alternative forms of photosynthesis. A tropism is a growth of a plant away or towards a stimulus. We talk a lot about phototropism, but there's other tropisms as well. Photoperiodism, we have flowering in long day and short day plants in preparation for winter. This is a response and change of length of the night. We have photoperiodism, again, that we have flowering controlled. We have long day and short day plants. Leaves change colors because of photoperiodism. Feedback loops help us maintain homeostasis. A really good example of a classic feedback loop is the release of insulin by the pancreas in response to glucose levels in the blood. Our immune response in mammals has a cell-mediated and a humoral response. Cell-mediated has the cells directly dealing with pathogens. The humoral response has B cells producing antibodies against specific pathogens. And antigens are recognized by specific antibodies produced by B cells to the antigen. In our primary and secondary immune responses, the secondary one is faster because those memory cells can produce antibodies much quicker, can recognize, it's more rapid, it's enhanced immune response. Okay, transformation experiment. You may encounter the results of transformation and be able to analyze each of the growth of each of the plates. And we did virtual labs and we talked about these a lot. If it uptakes the plasmid, maybe it'll have the traits that was incorporated into the plasmid. If it doesn't, figure out what logically is growing there because of what reason. Okay, so again, the plasmid, small circular piece of our bacterium. Be familiar with different experiments to talk about how we figured out DNA was the inheritable material. DNA replication, remember we have it going in the 5' to 3' direction. We have our leading strand and our lagging strand. Remember the enzymes that are involved. Remember the structure of our nucleic acids, our DNA. We have our phosphate group, our ribose group, our nucleotide base. In the cell cycle, we have G1, S, and G2, and there's checkpoints that lead us into each of these cycles. So again, we have cyclin and cyclin-dependent kinases. We have the MPF, which is our mitosis promoting factor. Our apoptosis is our programmed cell death. And remember, errors in each of those checkpoints can lead to cancer. Got 15 minutes. Back to reproduction. So, mitosis is going to produce 2 genetically identical daughter cells. Meiosis produces cells with half the normal amount of genetic material. Meiosis is the process that produces these haploid gametes. We have weird reproductive cycles as well. In fungi, for example, we can have a dikaryotic cell with 2 haploid cells fusing together. You don't need to memorize any of this, but you may be shown a cycle or something and just be able to encounter that and interpret the diagram. So independent assortment, random fertilization, and crossing over all give us better genetic variation in a population. Independent assortment is when these chromosomes are lining up in preparation for meiosis and they're splitting. We can either have them arranged in a certain order or in a different order, and that could give us different possible gametes. So in order to calculate the possibilities of these chromosomes, you just take the number of chromosomes and do it to the exponent. So for example, in 23 chromosomes, we do 2 to the 23rd, and then we get 8 million possible orientations. Okay. A random fertilization, you probably won't have to do that one, but our crossing over, we have functionally infinite combinations of information because they can cross over at any point with any gene on the chromosome. Okay, remember, genotype is the alleles that an organism has for a particular trait, and evolution, our definition, is the change in the frequency of alleles in a population. So these genotypes are what we're measuring in evolutionary studies. Phenotype is the actual trait the organism shows, like their fur, like their eyes. The law of dominance. So we have dominant and recessive genes in regular, complete Mendelian inheritance. Dominant traits are going to appear. Recessive traits, both alleles have to be in there. A monohybrid cross, you guys know how to do that. Remember, the parental generation is P, the next generation is F1, filial 1, the generation after that is F2. A test cross, also known as a back cross. You cross a homozygous recessive individual with your parent that you need to know the rest of their genotype. So if you're given, like, oops, of course. So if you're given a big T and you don't know, so you know this individual is expressing the dominant phenotype, you don't know what their second allele is, it could be homozygous dominant, it could be homozygous or heterozygous. You cross it with a homozygous recessive individual, and the results of that will show you what the other allele is. All right, we can have a dihybrid cross, and in order to figure out each of the things that go on the top and go on the side, you figure out the possible gametes from that individual. So if you had an individual that was big R, big Y, little y, think of all the possibilities. We could have a big R, big Y, we could have a big R, little y, we could have a little r, big Y, and we could have a little r, little y. And those are the 4 possibilities that would come out of that individual. In a crossover, if you have a crossover frequency or a recombination frequency of less than 50%, you can assume that the genes are linked, or it's a high probability that the genes are linked. If it's greater than 50%, probably not linked. Okay, as we're doing these, a dihybrid cross is going to come up in general if it's, you're actually doing this cross where it's 2 dihybrids, like this. You're going to come up with a 9 to 3 to 3 to 1 ratio. Not necessary to memorize, but it will save you some time. If you have two individuals that are heterozygous in each trait cross with each other, and you're trying to figure out if the actual phenotypes came up with the probability, if you know 9 to 3 to 3 to 1 for this particular cross, you don't have to do all the huge square. We worked yesterday with one that came out as 1 to 1 to 1 to 1, and that was because we crossed, I think it was a hybrid with a homozygous recessive individual, but that's easy to do as well. Okay, again, another dihybrid cross. The inheritance of unlinked genes are independent events, similar to multiple flips of the coin. So you multiply independent events. So for this one, Jordan, this is the one that you were mixing up yesterday, I think. We're going to cross, we want to see what's the probability of getting this whole genotype. So we're going to cross each of these potential combinations. So we take this one, and we just do a regular cross, and we figure out what the probability of getting this one is. So that's 1/2, so 1/2, and then we do this one, and this one, and this one, and this one. We multiply all those events together to get that particular ratio. Okay. It's chromosome map and crossover frequencies, recombination. So during crossing over, during prophase 1 of meiosis, genes can have these chromosomes meet in what's called the synapses, and they can cross over and switch places. And these linkage maps, we've practiced with those, we know how to do those. Okay, so some autosomal conditions, these are genetic disorders. Ones that you could use as examples. Huntington's disease, some that are autosomal recessive, Tay-Sachs, cystic fibrosis, PKU, sickle cell anemia. Just have a couple on hand as disorder examples. Chromosomal conditions, so we can have non-disjunction when our spindle fibers don't separate these sister chromatids properly, or it takes a whole chromosome to decide. Trisomy 21, Klinefelters, poly-X, any of those are fine. Okay, so you might be shown a karyotype. This is a karyotype, a picture of all our chromosomes, trisomy 21 with a female. Oh no. Okay, 10 minutes. This is our operon. You guys know how to interpret the results and what would happen with operons, right? So just make sure if you're given a diagram, you read it properly, you're able to explain something that is like actively repressed or something that is induced, it needs an activator or something else to actually cause transcription to happen. Okay, so gene expression and control happens on many levels. We can have it happen pre-transcription with our remodeling of chromatin, where we can expose certain parts of that chromatin or kind of make it hidden from our transcription factors. We can have it happen at transcription, whether or not there's a repressor on our genes. We can have it happen at post-transcriptional level. So when we get mRNA, we can add a 5' cap or a poly A tail to make it more stable. We can have RNA processing where we splice out our introns, or we splice in certain exons. We can have it maybe blocked to go into the cytoplasm, or maybe the mRNA is degraded before it even gets out of the nucleus. We can have translational modification. We can have post-translational modification where we get cleavage, chemical modification, or degradation of the protein by certain proteases. Cell communication. The signal is composed of generation, transmission, and reception. We have really complicated signal transduction pathways. We can have unicellular signaling pathways or multicellular signaling pathways. So it's categorized by a signal, a transduction, and a response. They are specific and regulated. One signal molecule can cause a cascade effect, releasing thousands of molecules in a cell. These pathways evolved from a common ancestor, so it's another mechanism that we can use as evolutionary evidence. We have autocrine signals, synaptic signals, paracrine signals, and endocrine signals. Remember, endocrine go really far. We have ligands, receptors, and then the signal can cascade within the cell and generally result in something to do with the gene. So whether or not we're going to block expression or increase expression. We have G protein linked receptors, so we can have our G protein catalyze enzymes. We can have ligand gated ion channels, so when that signal molecule binds to the protein, the gate is closed and then the ions can flow in once it has that conformational change and it's open. We can have a cellular response, most likely gene expression, or it can increase protein activity, or we can initiate apoptosis. Those are just some examples. Remember we talked about this cascade can get exponentially larger, so they can be really, really complicated, but they cause a lot of important pathways in our cell. So second messengers, remember, these are internal signaling molecules like cyclic AMP that can activate various metabolic pathways. We can have alterations in our signaling pathways like diabetes, Parkinson's syndrome, cancer is a big one too, antihistamines, or birth control. Pheromones are messages between individuals of the same species. Signaling pathways still work for these, but pheromones are important. FSH is an important one to think about, follicle-stimulating hormone. We graft the female menstrual cycle in response to different hormones. We have steroid hormones produced from cholesterol. These are fats. Steroids can go through the cell membrane. Non-steroid hormones need a receptor, so these can be protein hormones. Remember the structure of our neuron and talking about cell communication. Remember our action potentials and what's happening along the membrane. We talked about this one already. Remember that action potential is propagated down the neuronal axon, and then when it reaches the terminal, it can be have whatever neurotransmitter released into the next neuron. Viruses are not living organisms. Do not make that mistake. So they're dependent on other cells. They're extremely, extremely tiny. They are not cells. They need hosts because they lack enzymes for metabolism. They lack ribosomes for protein synthesis. They need the host machinery. They use the same metabolic pathways as the host, so they sort of borrow their metabolic pathways. We have different types of viruses. They're a vector for information. We can have lysogenesis in a bacterial host. We can have transformation. Sometimes viruses can be packaged with DNA from the host genome. That's called transduction. We have lysogenic and lytic cycles for the virus. HIV is a retrovirus. Make sure you can explain like the case study of HIV and how it uses reverse transcriptase. Big idea 4. I got 5 minutes. All right. So nucleic acids. Remember their structure includes a phosphate, a sugar, and our nucleotide base. So G, A, T, C, U. Proteins are made of long chains of amino acids, and then they can fold into different conformational structures, whether or not they have subunits. Carbohydrates, you can recognize those by sort of like this hexagonal or pentagonal shape. Our lipids will have long carbon tails, and maybe it's a different head, maybe they have a glycerol. Amino acid structures has our side chain, our amino group, and our carboxyl group. Remember, proteins and peptides, they may use those interchangeably. They're polymers, and amino acids are their monomers, so they'll form up long chains. Structure of DNA, we didn't mention this the other day, but remember these are hydrogen bonds in between the G and C. There's 3 of them here, 2 hydrogen bonds in front of the A and the T. G and C rhymes with 3, that's a good way to remember it. But if a DNA strand has more G's and C's than A's and T's, it's actually going to be a little bit stronger because there's more hydrogen bonds that should be broken. So it may ask you like which strand of DNA would be more likely to break in response to this enzyme or whatever. Pick the one with more G's and C's because that's the stronger one. By a little bit. Okay. Be able to identify organelles and the various functions of different organelles in the cell. Environment can influence gene expression. This is an example of our fox. In certain temperatures, the proteins will either express this black fur or the white fur. In the colder temperatures, it'll be white for the fox to be able to camouflage in the winter. We have imprinting behaviors. We have a lot of behaviors. Some of them are learned, some of them are innate. Hibernation, another good behavior to know. Relaxed state of inactivity among endotherms. Some mammals release hormones that lower the metabolic rate. We have extended torpor. Remember, taxis versus kinesis. Taxis is goal-directed. You're moving in the direction of something. Kinesis is random movement in response to a stimulus. Think of our little pill bugs. Homeostatic behaviors. I forgot what I was going to say for that one. Just keep them in mind. We have, what was I going to say with this one? Oh, endotherms and ectotherms. So, endotherms in colder temperatures have to have a higher metabolic rate to make sure they maintain their internal temperature. Ectotherms in general have a lower metabolic rate, but will have a higher metabolic rate in higher temperatures because they're warmer and just the higher movement of molecules in warmer temperatures, that's a thing. All right, make sure you can identify parasitism, mutualism, commensalism, competition. Invasive species are a big deal, so make sure you can identify invasive species and their impact on local environments. Yesterday we talked about the rainbow smelt. This is a graph identifying the introduction of an invasive species that outcompetes the other one. Other human impacts. The loss of habitat from deforestation will likely lead to a loss of species diversity due to a decrease of available niches. Other big ones are urbanization, industrial agriculture, burning of fossil fuels, water pollution, and runoff. We have direct effects of human impact, and then we have indirect effects like acid rain, global warming, that kind of thing. All right. Yes. Okay. So importantly, over the weekend, make sure you review stuff that really, like especially from this isn't everything, but especially from this, if I flash something really quickly and you guys were like, oh, I forgot about that, or that, it's been a while since I've seen that. please review it. Take time to like look at those tricky topics, do a few practice questions, but do not cram. This is not cram time. This is relaxed time. When you are in a stressful situation, I'm sure you've heard other teachers say this, there's actually parts of your brain that shut down the critical thinking and the logical thinking part, so it's very difficult for you to do like math or logic-based problems and short-term memory recall as well, if your stress hormones are up. So the calmer you can get, the more sleep you have, the more relaxed you are on test day, the better you'll actually be able to do. Okay? So take some time to relax, make sure you go to bed early the night before. I like this quote from Albert Einstein, "The whole science is nothing more than a refinement of everyday thinking." If you bring your phone, just leave it off or completely outside the exam room. You guys know the drill. Please, please, please do not forget your 4-function calculator. Does everyone have one at this point? Okay. On day of the test, I will be here at 7:30. I will be in my classroom. I will bring some goodies, and you guys can check in with me before we go upstairs. So if you want to show up at 7:30. I will have 1 or 2 extra in case of emergencies, but please do not count on me. So I don't want you, like, again, the stress hormones, I don't want you freaking out on the way to school, like, oh my gosh, I forgot my calculator, I gotta turn around, I'm gonna be late. I will have 1 or 2 extra. I do not have 10 extra though, so please don't forget them, all right? And if, you know, you're coming to school, you can shoot me a quick email and be like, oh my gosh, Miss Reid, I'm driving, I forgot my calculator. We'll make sure that you get one. Number 2 pencils, blue or black pens, and that is it. I did it. All right, it's 9:35.
Perfect.
If you guys have questions, let me know. I have little goodies before you guys leave. And some of these are like half.
It's like what Mr. Jake did for us.